Okay, I think we should start now. It's precious time, and I'm really happy to welcome Chas Bountra and his team to give a long overdue talk for the DRI, where we could start to explore potential collaboration with the Structural Genomics Consortium, which is leading in Oxford. So it's a broad effort to get basic research to real drug targets and to bring them mature enough to get them of interest for industry to develop for medications. And they have really a very broad platform to do that. Chas has a fantastic history in translation. He has been vice president in GSK, has done there a lot of clinical developments very successfully, and then moved to Oxford to set up this Structural Genomics Consortium. I'm very happy that he accepted to come and talk to us. He's in the middle of the renewal of this very successful consortium. And I think there will be plenty of potential collaboration. So we have a quite busy program. There are several people talking from the consortium. We have questions at the end of the talk. So please hold them for the end. You can use the question and answer box, and Giovanna Lalli will manage those questions. So without any further delay, I give the word to Chas. Welcome, Chas, and we are ready to listen to you.
Well, Bart, thank you very much, and Giovanna, thank you for organizing this. I'm just going to talk for maybe 2 or 3 minutes and just try and give you a sense of what we would like to achieve this morning. So in Oxford, the people who are on this call are from 2 groupings. One is the Structural Genomics Consortium that Bart talked about. And then the second is the DDI, the Oxford DDI, the Dementia Drug Discovery Institute. Now, what we would collectively like to do is try and achieve 3 things today. So firstly, in the Structural Genomics Consortium, what we've been doing is, for the past 15 years, we've been working on novel genes, novel proteins that we think are therapeutically important. They've got strong association with human genetics, etc. And for those genes and proteins, we have been making early discovery tools. For example, we've been purifying the human protein. We've been building biophysical, biochemical assays for that protein. We've been working out the structure of that protein. We've been generating high quality inhibitors for that protein. So these inhibitors, they're potent, they're selective, they're cell active. And in some cases, we've generated antibodies. Now, these tools are very high quality tools. They're very high quality reagents, extremely well characterized. And the reason they're so good is not because we're clever, it's because we've been tapping into the resources of 10 large pharmaceutical companies. So they've been giving us access to their chemistry, et cetera, et cetera. So over the past 15 years, we've generated lots of these tools. And many of them have been focused on neuropsychiatric diseases. Many have been focused on inflammatory diseases, many for metabolic diseases, and many for cancer. Now, we believe many of these tools will be of use in understanding Alzheimer's biology or neurodegenerative disease processes, etc. So we would like to share those tools with everybody in the DRI across the UK. So that's one objective. The second objective is, Bart, that what we would like is, you of course are all the Alzheimer's experts, and you have new targets, new genes, new ideas that you're interested in pursuing. We would love it if you would nominate those genes and targets, and we will then do what we're good at, and we will take those and we will generate protein structures, assays, inhibitors, antibodies, etc. And then again, share them with yourselves and indeed the whole of the DRI to accelerate science and therefore accelerate therapeutic discovery. So that's the second objective. The third objective is the Dementia Institute, very much like what you've set up in University College and is also being set up in Cambridge. Under John's leadership and working with Eleanor and Paul, What these three individuals have done is, in the past 4.5 years, they've created a beautiful pipeline of new targets that they're pursuing for dementia. They've created a large network of collaborators. Importantly, I think John and Paul and Eleanor have been extremely successful at forming very, very strong partnerships with industry. And now they're actually thinking of partnering some of their targets and assets with pharma companies, but they're also talking to venture capitalists and thinking about how to create companies around some of these targets to enable translation. And so what we would like to do today is we would like to see the DDI working more closely with the DRI more generally. So three objectives. We want to share our tools with yourselves. We would like you to nominate targets, novel targets that we can generate tools for. And thirdly, we would like the DDI to be working much, much more closely, but with the awesome network of people that you've set up in the DRI. So three objectives. So
Fantastic.
On that note, I will shut up, and I think I'm handing over to Ofer, is it?
Yeah, so I'm unmute now, and I'll try to share the screen. Right, we we normally present this sort of roadshow over 2 or 3 hours, so this will be a very condensed version, and I hope we'll manage to leave some time for questions at the end. And so I'll present the SGC in general and a bit of my work in the context of an NIH-funded project, which is called OpenAD, and I'll explain exactly what this is. So really, the SGC's view of the world is that there are always, in any class of genes you look at, for instance, these are the human protein kinases, or 500 of them, there are about 10% of celebrities that dominate all of research and all of literature and all of the press. And so in the case of kinases, you'll see that if you rank them by the number of citations in PubMed, you'll see that the top 10% or so get 80% of publications, and there's very little publication on the trailing end here. And by other measures that look in an unbiased way, such as sensitivity of cancer cells to RNAi or driver mutations, they are distributed all over the kinome. So it's not that these are more interesting, but there are a lot of reasons why you can get more funding and more interest in things in which there's already publication and some knowledge. And we're trying to break that mold in working on the tail end, on the interesting targets that are not widely investigated. And so the SGC has been founded 15 years ago, 16 years ago, actually, and I'll put a few bullet points of what it is that we do and what is our competence. So primarily, we promote development of new medicines through open access research. Everything we do is deposited in public databases or published or on our website very soon. And we focus on less studied areas of human and pathogen biology. We don't trust ourselves entirely to nominate which target we work on. So we work within very extensive networks of experts and collaborators that help us to choose which are the targets we should work on, and also to utilize the output of our research. Our key competence is around protein-centric tools for new targets. So we're talking about purified proteins, crystal or cryo-EM structures, biochemical and cellular assays, and we're looking at to validate antibodies. As you know, many commercial antibodies are not really up to speed. The SGC has been a world leader in discovering structures of novel human proteins. We have a sizable percentage of the protein data bank. So that's been our reputation for many years. But we've since evolved into producing what we call target enabling packages, which are packages of data and reagents that are solid foundation for medicinal chemistry. And I'll talk more about this. We've been developing chemical probes in collaborations with chemistry industry and also in-house. These are drug-like molecules that can be used to generate and test therapeutic hypotheses in cells and organisms. And I just put this on the slide. I won't go into this. It's very broad target areas. We have expertise in protein kinases, epigenetic modulators, and so on. But beyond these areas, we have been looking at a very diverse set of individual proteins that are associated with disease. When they come up, when there's interest, we have the tools and the means to move forward on those. So this is a busy slide, but it indicates the process of the concept of target enabling package and where we sit in the larger ecosystem of developing drug targets. So identification of drug targets comes from a lot of different routes, from genetics, transcriptomics, model organisms, etc. And there are always questions about causality of these genes, directionality, what happens when you modulate these genes or these activities, and the integrated response, side effect, and drugability. And then at the tail end of this, there is the development of chemical probes of very specific tool compounds that allow to investigate physiology, but also lead optimization into clinical molecules. And we feel, and many others, that there is a gap here in the middle between the people, the labs, the expertise in this area, and the expertise in this area. And this is really reducing this to practice to say, well, there's a gene that's involved. Let's see what happens to the protein. How is the protein altered by mutations? What is the activity of the protein? Can we develop assays that are useful for deciphering function and moving towards medicinal chemistry? Structures of protein have been very helpful in supporting medicinal chemistry and other things here. So the areas that are shaded in red are what we focus on doing. And the rest is usually through our collaborative network. And I would say that we really use the open access aspect to crowdsource research. We put these reagents out there, people will come up with unexpected ideas and results. The OpenID is a new project. It's a 5-year multi-center consortium funded by the NIH, or National Institute of Aging rather, to discover new medicines for Alzheimer's. And the idea is to move away from the targets that everybody else is working on. So we're not looking at amyloid, we're not looking directly at influencing tau, we're trying to mine the data for other targets. And this is one paper that gives an example of how they use proteomic analysis or transcriptomic analysis from brain sections from people with advanced, mild, or no disease that are age matched. And these are many hundreds of brains were used in the analysis. You come up with clusters or modules of genes that some of them are higher in the hierarchy. There's a hypothesis that they may, you may modulate them and drive desired consequences in disease. And then so you scale them, you score them by disease relevance, tractability, and then what we do is we try to fill up this profile with all the ingredients that are needed to do research. And there are different cores in Oxford, in Emory, in the University of North Carolina, and in Sage Bionetworks, and many other universities. It's a very big project that we are about 10% of. I will skip this. You will have the presentations later, but this gives you an idea of what kind of targets we are working on. That's a list from a couple of years ago. Some of them may be familiar to many of you, but others are really things you've never heard about. And the type of output we deliver on these is, for instance, we've been looking at the interaction between CD44 and its external binders and its internal binders, which are firm domain proteins. So we look at the firm domain proteins, we have crystal structures of where CD44 binds, and we use crystallography to identify molecules that can interfere with that. On the outside, and assays for high throughput screening. On the outside, we're looking to try to inhibit its interaction with outside molecules. Here we've teamed with DNA encoded library company, XChem, and we have some hits that we're working on now. I will end by a couple of slides from Brian Marston, which we show how we document and disseminate the data that we generate. Other than normal publications, we capture every data point, every experiment in an electronic lab notebook and structured database. These are then the data that go into a target enabling packages, go on our website, and they're shared on Zenodo, which is a stable website with DOIs. So those documents are searchable and remain there forever. And we have joint infrastructure and collaborations with other groups that look at target biology. And just to blow off your chairs, this system is really the envy of the world, or anybody who's heard of it, because we really record every experiment in full detail. You can go back to an experiment from 14 years ago, search, find it, and get all the details, the gels, the mass spec, and the data. And so my time's up. I'll end here, and I'll just show quickly the collaborators, and you will hear from some of them later in the day. And the OpenID and TreatID are the consortium in the US that we're part of. I suggest that we leave the questions to the end. And now, Paul, would you like to share your screen, or shall we continue this way?
You can continue that way. I can just say next slide.
Okay. Here we are.
The screen is not shared yet.
Oh yeah, I'm sorry. Trying to simplify things, make them worse. Yeah, here we are.
Yeah, fine.
Okay, there we go. So I wanted to describe the work we've been doing around chemical probes and show a specific example where it applies to a neurodegenerative disease. So the chemical probe project really sprang out of all the work that had been done that Ofer described around the structural biology of novel targets. And the question we had about a decade ago was, you know, when you have protein, you have crystal structures, what else can we do that would generate value from these reagents, especially for understudied proteins? And so the idea was that we would develop small molecule chemical probes that specifically inhibit members of these protein families with some selectivity and cellular activity, and these probes would be used as tools to understand the function of these in cellular assays primarily. So we define a chemical probe as a molecule that has potency of 100 nanomolar in some biochemical assay, usually an enzymatic assay or potentially a binding assay. It also needs to have some selectivity over similar proteins, and we think that 30-fold is a good compromise between feasibility and finding molecules that have 30-fold selectivity and also utility. So if we test these in cells and we see an effect, we have pretty good confidence that the effect we see is due to the annotated targets. And then finally, these compounds have to work in cells, and so we want to see a cellular activity in the micromolar range, which is not too far from the activity we would measure on recombinant proteins. So again, we have confidence that the effect we see in cells is related to the target we think it is. So that is our definition of a chemical probe. The process of making these is shown in this chevron plot below. We start with the protein that was usually developed for a crystal structure, and then we use that protein for the developed assays, and these might be binding assays or functional biochemical assays. And then with that assay and that protein, then we try to find a hit compound, which is a small molecule that would modulate the target. And there's a variety of methods we use for that. Virtual ligand screening, which is purely computational, followed by testing, or fragment-based drug discovery, which is a technique very well suited to the structural biology capabilities of SGC. But we also use high-throughput screening in collaboration with a number of big pharma partners. Once we find a hit, that compound itself is usually not good enough to answer any biological questions, and so it needs to be optimized into a compound which is more potent and usually more selective. And so that's where we employ sort of standard medicinal chemistry, structure-based drug design techniques to design, synthesize, and test compounds in a hit optimization stage. When we achieve a compound we think meets our criteria, then that compound is characterized extensively. In fact, some of the SGC chemical probes are the most well-characterized small molecules that I know of. And so we look at broad selectivity panels of proteins in the same family, whether that's kinases, looking at as many kinases as we can, or some of the energetic ones I'll show you in a minute. And we also look at broader drug selectivity panels. There's a number of commercial organizations like Serup and Eurofins that will do broad screening. And so we have a general idea about the selectivity of these compounds. And then again, we test it in cells to try to show an on-target effect. We can actually affect the target in a cellular context. And if anything about the biology of the target is known, then we'll also look at functional assays in cells. With all of this characterization done, if our compound meets probe criteria, and we have an external committee that looks at the data and gives their seal of approval on our chemical probes, then we publish this on our website. And Ofer just described how we would do that, but we also try to publish this in a journal as well, because then we get a sort of worldwide press for these chemical probes. And then rather than just a package of information, we want to provide these compounds as physical samples as well. And so we scale up enough of the compound to distribute it to collaborators, and we work with standard commercial chemical suppliers to sell the compounds at a reasonable price, either as single compounds or as entire sets to use in cellular screening. And then with all of that probe work done, these compounds then go out very widely into the scientific community and are used to really discover novel biology, and in some cases find some exciting drug targets as well based on the chemical probes. Next slide over. This effort is really a global effort in the SGC, which has the support of a number of academic groups as well as large pharma partners. And so all of the big pharma shown on the left-hand side both fund and also work collaboratively with the SGC to develop the chemical probes. And it wouldn't really be possible to develop all of the chemical probes that we have using just internal medicinal chemistry. And so what the pharmas bring to the table is a huge amount of medicinal chemistry expertise and resource to make lots of compounds as chemical probes. They also bring to the table things like high throughput screening technologies and other hit finding technologies that we struggle to access on scale in academia. The screening of the compounds generated by the pharmas is done internally in the SGC, where we have a fantastic assay group led by Oleg, who's on the phone today, as well as a number of cellular assay groups as well, and Killian may mention that as well. And we also do the crystallography of the molecules along the way in the chemical probe discovery process to enable their optimization more quickly. And I also mentioned that we have a number of academic collaborators. There's SGC sites all over the world now. We work with chemists at all those organizations in our chemical probe project, as well as more specific ones. We had a great collaboration with Institute of Cancer Research in the UK and all of our friends in Oxford as well to help develop these chemical probes. So that's the sort of general outline of the probe project. If you go to the next slide, I'll give just a sort of snapshot of where we are now with our sort of chemical probe library. Over the past decade, we've developed 50 probes. This is a joint effort with all the pharmas I mentioned, as well as our academic collaborators, and also the SGC site in Toronto, where we have a large group working on chemical probes as well. And the area that we initially focused was in epigenetic proteins. So there's a number of epigenetic processes, and we focused really on histone modification, because when we started working on this, there was hundreds of both writers of histone marks and the erasers of histone marks and readers of histone marks as well. And we thought that the chemical probes for these epigenetic proteins would be a fantastic way to really discover what are the interesting targets in this space. Ofer is confusing me by messing around. And if you sort of just look at what we've done at Oxford, we've really focused on the readers of lysine acetylation, the bromodomain and Yeats proteins, as well as some of the readers of lysine methylation. So this is just a picture on please.
1 minute, please.
Okay, thanks. I'm talking too slowly. So this is just a picture of the protein families we've worked on the right-hand side, showing their success in bromodomains and methyltransferases. Next slide. So in the remaining 50 seconds, I'll just describe very quickly a project that came out of this chemical probe efforts. And this was looking at a disease called Friedreich's ataxia, which is a rare disease that's caused by a trinucleotide repeat in the gene for this protein, which is frataxin, which is an essential mitochondrial gene. So if you have this trinucleotide repeat, the level of frataxin is significantly reduced to the point that it causes neurodegeneration of spinal cord. It leads to this ataxia. But in heterozygotes, there's actually have no functional effect by having reduction of the protein. So we wanted to find a way of increasing the level of frataxin at least to 50% of normal. It was known that there were some genetic alterations caused by the trinucleotide repeats, which appeared to center on histone 4, lysine 20 methylation, as well as histone 3, lysine 27, and lysine 9 methylation at the splice sites of the protein. So it's a collaboration with Richard Wade Martins in Oxford, who developed a luciferase reporter assay to screen for compounds that would increase the expression of frataxin in HEC293 cells. Next slide. I'm not counting the time it takes to change slides, Gopher. So this was a screen that was done that Richard and his student Gabriella did, where they took our library of epigenetic compounds and screened them to see changes in this luciferase-linked frataxin. And so that's the graph shown on the left. And what was interesting is a number of histone methyltransferase inhibitors actually increased frataxin expression up to near wild type levels. One of them that was most interesting was this compound A196, which is an inhibitor of two lysine methyltransferases called Su420H1 and H2, which are responsible for methylation of lysine 20 on histone 4. This was a probe that was developed in collaboration with AbbVie and University of Toronto. So what was also surprising of this is this compound not only did it increase the levels of frataxin, but it seemed to do it quite specifically. So if you look at the RNA-seq of the cells, you can see there's only actually 30 genes which are modulated by A196, including frataxin, which is the most modulated, shown in the red circle. So it's a quite specific effect. So we got funding from the LifeArc Philanthropic Fund, and an excellent student of mine, Rob Quinlan, has been working on the structure-based design of optimized versions of A196 to improve the potency of this and test these types of compounds in animal models, which is the analog shown in the top right. Okay, now we'll wrap up there, and I guess we're having questions at the end.
Okay, so my name is Elena Di Daniel. I'm the head of biology at the Oxford Drug Discovery Institute. So we are one of the 3 DDIs that are funded by Alzheimer's Research UK, established in the last few years to translate novel findings into a cure for dementia. The intent is to translate cutting-edge academic science into drug discovery and to prosecute projects from target to lead status and beyond. The ODDI operates as a highly interconnected network of collaborators from academic laboratories to pharma, nationally and internationally. And together with OFFER, we are collaborating with the OpenAD NIH-funded team to test innovative molecules in our well-validated cellular assays. OFFER and his team, and more broadly, the SGC, are key collaborators for the ODDI. First, providing chemical probes, and secondly, through structural biology support to facilitate drug development. And so in the next couple of slides, I will provide some top-line examples of some of our projects. As a strategy to enhance clearance of protein aggregates in dementia, we are investigating lysosomal functions. We have utilized the SGC chemical probes, such as a Picas kinase set, to identify kinases that regulate lysosomal functions by measuring TFEB translocation, which regulates clear genes, and using DQBSA as a readout of lysosomal activity, utilizing a high-content imaging platform. In a collaborative effort with Professor Wade Martin's laboratory, we have identified a number of interesting compounds that positively regulate lysosomal activity and are effective in clearing protein aggregates in vitro, as demonstrated by our colleagues at the Alborada DDI. The SGC expertise in chemogenomic target ID is proving useful when we have also screened libraries of poorly annotated compounds that resulted in the phenotype we were interested in. And thanks to an AR UK network cooperation grant with Katie Lannon and colleagues at Exeter University, we have been investigating epigenetic marks in iPSC-derived neurons and macrophages treated with the SGC epigenetic probes, creating a data set that will be interesting to integrate with DNA methylation and RNA sequencing data from human Alzheimer's brain. The next slide, please. As a human Alzheimer's disease genetic has pointed out to TREM2 signaling pathway, we have initiated a number of parallel approaches to identify the best point of therapeutic intervention. Capitalizing on the Oxford iPSC macrophage expertise in Sally Cowley's laboratory, we are routinely utilizing isogenic iPSC-derived macrophages to study the consequences of gene mutations of TREM2, PLC-gamma2 on intracellular signaling, as well as on cellular functions such as phagocytosis, migration, cytokine release, etc. We have devised biochemical screening assays for PLC-gamma2 and SHIP1 that we are now utilizing in high-throughput screening campaigns to identify 2 molecules that can be utilized in vitro assays and ultimately in vivo proof of concept studies. A joint PhD student with the SGC and ASI has recently obtained TREM2 crystal structures in complex with single chains variable fragments and currently being deposited in the PDB. And she's currently studying the modulation of TREM2 shedding and intracellular signaling using the ODDI iPSC platform. In addition, we are excited to be collaborating with Peter Hislop and with Bicycle Therapeutics to develop bicyclic peptides targeting neuroinflammation in the brain, an approach that could combine the antibody specificity with small molecule size. Next slide, please. So moving on to Parkinson's disease, which is another common cause of dementia, I'm now going to briefly highlight the approaches that we are taking to identify targets and prosecute them through drug discovery. Benefiting from academic target nomination and the SGC target prioritization pipeline, ultimately resulting in the generation of target-enabling packages, we are developing a platform of assays that is allowing us to study intracellular dysfunction at the mitochondrial, lysosome, and vesicular trafficking level to validate candidate targets. The most advanced project we're working on is a collaboration with Celgene and with the Oxford Parkinson's Disease Centre, or PDC, to develop a novel treatment for neurodegeneration. The pharma brings in its medicinal chemistry resources, while Oxford offers a fantastic disease center platform consisting in longitudinal clinical Parkinson's disease cohorts, in vivo and in vitro disease models, allowing human-focused target validation and compound testing. New target classes that we want to study in the future include RAB GTPases, lysosomal solute transporters, 2-port channels, E3 ligases, each of which are also targets classes of interest and SGC expertise. So do feel free to reach out if you wish to discuss more, and happy to take any questions at the end. Thank you.
Thank you.
Okay, thank you. We'll move now to Liz Carpenter. Are you online?
I think so. Can you hear me?
Yes.
Hello?
Yes.
Good. Okay. So I'm Liz Carpenter. I'm one of the membrane protein structural biologists. I'd like to talk about some of the more difficult targets that we work on at the SGC to give you the impression it doesn't have to be a small domain of a protein. We can look at large complexes. Could you move to the next slide? Yeah, so we work routinely with large complexes of proteins. We also work with integral membrane proteins, and on the next slide, my favorite, which is large complexes of integral membrane proteins. So on the next slide. So just to say a bit about how we produce very large complexes, this is work by Nicola Burgess-Brown and Wyatt Yu. Expressing proteins in multi-cistronic systems. I think Nicola's gone up to 13 proteins in one. Wyatt's looking at a series of methods to produce proteins for cryo-EM, including knocking in tags into the protein in cell lines that express the protein, making antibodies, and then making large 8-component complexes with expressed protein, which can then be solved to high resolution. I think that one went to 2.4 angstrom by cryo-EM, so very high quality structure there. And on the next slide. Are we moving forward?
Yes.
Okay, it's not come up on my screen. So just to say, okay, so I'm not, okay, good, it's come up on my screen now as well. So in terms of exactly what I mean by membrane proteins, I've realized that sometimes cell biologists don't think of things the same way as protein biochemists do. So we generally work with proteins with a very large number of transmembrane helices. So we're happy with anything from 2 to 20 transmembrane helices. We're finding increasingly these form complexes with single pass proteins as well. So we need to be able to produce both proteins in the complex to get a functional, active, well-behaved protein. That gives us about 3,000 targets. There are 2 groups at the SGC who work on membrane proteins. We both work in Katerina Dua's group and my group. We both work on ion channels, solute carriers, and enzymes. I cover the ABC transporters, and Katerina covers tetraspanins and a large range of other sort of single membrane proteins as well. So if there are any membrane proteins you're working with that you'd like us to help you with, just get in touch and we'll let you know which of our groups look at these things. And we use cryo-EM, X-ray crystallography, even XPEL to solve structures. Next slide, please. So just to give you an idea of the sort of throughput, people think the membrane proteins are challenging. Years ago, we used to produce 1 structure a year. Then we moved up to 2 structures a year. In fact, we've also produced, Paul Brennan described small molecule probes, so we've also produced small molecule probes. Last year, we had 4 structures. This year, we were hoping to continue the exponential growth, but the virus may have got in the way. But nevertheless, we have several new structures coming through as well. So on the next slide, relevance particularly to Alzheimer's disease, I've just pulled out some genetic hits. Although we don't have structures of the actual genetic hits, we do have structures of close homologs of many of these. So for example, FIC1 or KCNK13, we have structures of 3 of the homologs. For the ABC transporters, I'm particularly interested in ABCA7, and we have people, I have a student who's producing that at the moment. Well, I have a student who would be producing that at the moment if the labs were open. But we have produced 2 structures of ABC transporters and mitochondrial ABC transporters in this case. In fact, we produced the first human ABC transporter structure. Similarly with solute carriers, we solved GLUT3, 2 members of the EAT family, and another transporter as well. So we have quite a bit of experience. So if there are transporters, ABC transporters, ion channels you're interested in, or even enzymes as well, please let us know. Okay, so on the next slide. So in terms of working with membrane proteins, we don't just solve structures. In fact, solving structures is probably 30% of what we're interested in. We aim to have a structure, crystallography or cryo-EM, but the target enabling packages that I think Ofer talked about, where we have assays, bound spore molecules, and in some cases, very interesting proteins. The ones we're particularly keen to look at are the ones where nobody knows what they do, and we're very happy to go out there and work out what they actually do. Part of what helps with that, of course, is having small molecule probes, and we have produced 1 small molecule probe. We're now, through the EUB Open grant, we're now funded to develop a whole series of small molecule probes for solute carrier proteins. So if any of those are of interest. And we have found that for membrane proteins, actually making antibodies, or nanobodies in this case, that bind to the protein. In the case of some ion channels, for example, we can see we can actually regulate the function of the ion channels. In this particular case of TREC2, we can both activate and inhibit the protein with different antibodies. So it's and different nanobodies. So it's an alternative route to finding a way to regulate the function. And of course, if it's at the plasma membrane, that can be added to cells as well. And on the last slide, I think my last slide. Yeah, so this slide is just a summary slide to show one of our target enabling packages for one particular ion channel. So in this particular case, task 1, we have 2 inhibitors provided to us by Bayer. We have electrophysiology. We showed the binding site of these compounds, and very curiously, we ended up with a totally novel way of regulating an ion channel. We have this little cross structure here that is actually controlling a channel that is not normally gated in most members of the family. So that story is hopefully coming out in Nature very soon. So that's the sort of information. If you have a protein you're really keen to work with, maybe you've got cell biology and so on, we can provide structures, binders, small molecule probes and so on, selective small molecule probes, which will be useful in cells. Okay, so that's where we are in terms of both large complexes and membrane proteins.
Impressive.
Thank you, Liz. And so we will move to Kilian Huber of the Chemical Biology Group.
Thank you very much, Alpha. So my name is Kilian Huber. I head the chemical biology team at the SGC and TDI in Oxford. And we are very much interested in using chemistry to study the biology of new protein targets, as mentioned earlier. And here's one example of a protein family that we have been working on for the last couple of years. And these are the so-called DEN domains, differentially expressed in normal neoplastic tissue. So they were originally associated to cancer development, but it's been shown over the last 10 years or so that these proteins the majority of them seem to be guanine nucleotide exchange factors for RAP GTPases. And this is still a largely unexplored family of GAPs, which interestingly has multiple links to many different diseases. The most famous one, DEN domain containing protein of this is probably C9orf72, which is, as many of you will know, causes ALS and FTD. But there's also other interesting family members, as in there's, for instance, the DEND2A, which has also been linked to mental retardation. There's DEND4C, which is linked to GLUT4 trafficking, which is relevant for diabetes. There's also recently was a structure for folliculin that is causing Birt-Hock-Dubé syndrome, which is also a malignant skin disease. And then there's MAD, which has also been linked to Alzheimer's disease, and many more. And on the right-hand side, you see what we have been doing so far. So as has been alluded to in the beginning, is at SGC, we like to focus on the whole protein family because this is very efficient, as in once we know how to express the proteins, we like to get as many structures as possible, because even though most of these proteins obviously share a common DEN domain motif, they have additional domains, and you learn a lot from additional structures. Plus, it is very enabling to have all these proteins, or the majority of them, in-house if you want to do small molecule inhibitor development, because then you also have a panel that you can use for cross screening, selectivity screening of compounds. So these DEN domains facilitate the GTP or GTP exchange from GTPases, RAP GTPases. If you go to the next slide, please. Thank you. So we have actually not only been solving structures of the DEN domains, so the GAPs, but also DEN domains in complex with RAP GTPases. So one of them is DEND1A in complex with RAP35, but we're also working on the RAPs independently or separately. And recently we have solved new structures for RAP39B and RAP38 in complex with either GTP or GMP-PNP. RAP39B is also a very interesting protein because it has also been linked to early onset Parkinson's disease, and is also thought to be a binder or intact of C9orf72, and also linked to ALS. And also RAP38 mutations have been reported in FTD. And so what we are currently trying to do is try to see whether we can use these crystal structures to understand how we can make potential inhibitors, both specifically for the GTPases, which has become more established recently, if you think about the advances that have been made about targeting KRAS. So we think there's a way in here, but we're also particularly interested in targeting the GAF GTPase complexes. And to that end, we also employ fragment screening, but also cellular screening, chemoproteomic screening, to identify starting points which can then be developed into chemical probes that enable the further study of the function of these proteins, both in physiology as well as in pathophysiology. If you go to the next slide, please. So this is on top of what we do with respect to development of chemical probes, or I should say it's actually complementary, because we want to use these chemical probes to study the function of proteins inside living cells. Selectivity is a key challenge for us and an issue. And because we work with living cells and with recombinant protein panels, you can only cover a few hundred of proteins, like, the kinases, GPCRs, etc. So for us, it's very important to understand how compounds actually, or which proteins compounds bind, or chemical probes bind, and drugs bind inside living cells. And that to that end, we use a target deconvolution platform that we have established here at Oxford, which is mainly focused on proteomic approaches. Of course, this can also be used for phenotypic drug discovery, and we are working with a number of partners, both pharma as well as charities, where we look at small molecules or drugs that have interesting phenotypes and then use our platform to identify the cellular targets of these compounds, which then hopefully will enable compound optimization, as well as biomarker identification for clinical trials. And as you may know, most compounds or most drugs actually that are approved are thought to interact at least with 6 known targets. So there might be many more that we are not actually aware of. And just to illustrate it a little bit at the bottom, I'm showing an example of a cytotoxic drug called gemcitabine. It has multiple targets that are relevant for its mechanism of action. So it inhibits ribonucleotide reductase, which is the green protein shown in the center, but gemcitabine depends also on the activity of a number of other proteins that it binds to, like solute carriers that have been mentioned by Liz, and kinases, which are required for uptake, metabolic activation, and so forth. And ideally, you'd want to know all about these targets to be able to understand the mechanism and also design clinical trials. If you go to the next slide, please. Yeah. And this is just, on this final slide, I'm just highlighting the different approaches that we are using. So we are mainly focused on proteomic approaches because as we are interested in proteins, these approaches allow us to measure direct biophysical interactions between ligands, as in small molecules and drugs, and proteins inside cells. And we use classical drug affinity chromatography as shown on the top left, where we immobilize ligands or append a function handle to a small molecule that has an interesting phenotype. And then we use that handle to immobilize the compound and pull down binding proteins from cell lysates or also intact cells, depending on the chemistry or approach we're using. So for instance, with click chemistry, we can also look at intact cells. We also use photoaffinity labeling for low affinity targets and ligands that tend to interact with membrane proteins. And last but not least, the latest addition to our toolbox is thermal profiling, which is an approach that we published in 2015 together with other groups as well, which takes advantage of the fact that ligand binding induces a conformational change in a protein, which can be measured as a change in thermal stability. And this obviously has been used for structural biology for many years, but we can now also use this to look at the binding of drugs inside cells. And what we do is basically we treat the cells either with vehicle or the drug and then heat the cells in the PCR machine. We lyse the cells and then use quantitative mass spectrometry to quantify the abundance of proteins in the lysates after centrifugation. So the idea is that once a ligand binds and stabilizes its target, you can recover more of that target compared to your vehicle treatment from the soluble fraction with mass spectrometry. And we can do this in a one-dimensional format where we use one high concentration to look at all the protein targets, or we can do a 2D thermal profiling approach where we use multiple concentrations of a given compound and across a given temperature range. And the beauty of that approach is that it doesn't require chemical modification of the compound of interest. So you can take it out of the box as it is, order from Sigma and do your experiment in 30 minutes later if you have the cells ready to go. And again, it can be, this approach is compatible with both cell lysates as well as intact cells, which is very enabling. Thanks very much, and I'm looking forward to taking questions later.
Okay, thank you, Killian. And then I think I'll hand over to John Davis, who has a summary slide. John, are you on?
Switch on your microphone. John? John, we don't hear you.
Hopefully you can now.
Much better.
I don't want to say too much because I think you've heard some really good examples. Just wanted to make a couple of points in summary. And some of that comes from my view as a customer of the SGC. So on the one hand, over on the left hand of this summary, you can see you've got the OpenAD consortium, which Opa's talked about. And you could also consider the DRI as a group a bit like OpenAD. And this is because we need fuel to feed the SGC engine. We need the nominations and we need the disease experts and their appraisal of which targets are really the most interesting. And then you've heard a lot about what the SGC can deliver. And I've rather crudely just summarized that as the TEP output. The TEPs are just sort of one package of particular types of product that the SGC produces. But I think you've been able to see from the presentations that have been given that you can access clones, obviously, that are used to make expression constructs and expression systems. There are structures that you've seen, there are antibodies, there are loads of assays, perhaps more than people realize because of all the selectivity screens that are run in Oleg's group. And then ultimately there are these molecules. They might be just original hit matter, or they could actually be molecules that have been worked up and are probes. And Paul explained that a probe is really quite a well-defined, well-characterized, potent and effective molecule that can be used in biological proofs of principle. And then on the right-hand side, you've got all the customers. So you're probably aware that SGC puts out all of its data so that everybody can use it. So the global community benefits from access to SGC product. Groups like the DRI, and I think this is really important, real value comes when there is biological impact as a result of people using the SGC tools. And Liz just said a few minutes ago that in fact, only 30% of her work is really structural work. The rest is around all of the assays and the biology. And this spreads on out into the biology. So it's really good if you've got somebody inputting on the left-hand side, but also ready to pick up on the right-hand side. And that leads to rapid impact if there are people waiting for the product for an interesting gene. And that's the key message I want to get across. This is a collaborative enterprise. The SGC is a really powerful hub in the middle, but it needs input, and then it needs people to use its output. And as a DDI head, I'm definitely one of those. I love it when there's a target we can work on that the SGC is engaged in. The quality of the product that the SGC delivers is second to none. And I remember a pharma colleague who is engaged with the SGC from Pfizer, and he said he likes it because he knows that when he takes an SGC product out of the freezer, he's very confident that it's going to work. He can rely on the SGC output. So that's, I hope, was just a quick summary, and also to bring us back round to why we're talking to you. We need basic scientists to have ideas and input and bring forward interesting targets and effectively prioritize them so that we are looking at the new genes that could be those ones of interest, because otherwise there's an awful lot in that original kinase curve that Ofer showed you at the beginning. There's an awful lot we could choose from, but there's very little literature for. But if there are new things that have been highlighted by studies in the basic sciences, then we know that it's more likely to create an impact down the road. And I think we should go over to questions so that we have some time for that.
So while they are accumulating the questions, I want first of all to thank you very much for this comprehensive, short but very effective series of presentations. It's very impressive, and I like it also very much. This bringing together of a lot of expertise, which is really getting all the tools together to get your biological question and your therapeutic hypothesis tackled, which we usually takes years for a lab to build up all this expertise around their protein. And so you could gain a lot of time by just working with you. So a question which came up with me is how does this work in practice? So one of our investigators is fascinated by a molecule, an enzyme, a gene, and wants to work on it. And how does it work from there? Can he just call Chas and say, I want you to work on this, or how does it work?
Brian, do you want to have a go at that one?
Yeah, so we're always open to nominations and ideas, and I think one of the best ways to let us know is actually just to reach out to us by email. And we do have nomination forms that we can ask people to fill out. But to be honest with you, it's best if you just talk directly to us, and we can then direct you to the right person who has the expertise in that area. So yeah, don't be shy. Please contact us.
Well, Bart, of course, it may well be that we're already working on it. It's already in our pipeline. So I think John summed it up nicely, actually. I think I hope you've all got the impression that we want to work on completely novel ideas, novel genes, novel targets, etc. And Liz pointed out we quite like doing things that are difficult and people think are intractable, etc. And I think the other thing is that I hope you've got a sense that we are extremely collaborative. Because we share everything, we're happy to make everything open, we want to release everything immediately, we're extremely collaborative, and so we're working with lots of people across the world. And I think John said it nicely. I mean, we're almost a sandwich between the ideas and the impact. We need you to come up with new ideas, we will then generate the tools, and then we'll work with yourself to create impact. So,
yeah. So to the audience, if you have questions, please use the question and answer box, and then Giovanna will lead the question to our speakers. So please use this opportunity to ask whatever. So I wanted to ask also something about this open access policy and how you achieve that. It's also something which I would like to install in the Dementia Research Institute. I think we need to go beyond the classical publication, and I think bioRxiv is one step, but I had the impression that you are a little bit more revolutionary even. Did I understand very well that your electronic lab notebook is freely accessible for the outside world, or is that a bit an overinterpretation?
I think Brian's getting ready to respond to that.
Yeah, no, so we don't make our electronic lab notebook open to the outside world, mainly because often what we've written needs to be interpreted in the context of the science that we were doing at the time. If we make it openly available as it is, it can be misinterpreted. But again, I would just repeat what I said before. If there's something you're interested in, contact us and we can make what we have available to you. There's no constraints upon that.
But what do you mean then with this completely open access and everything freely available? If you go to your webpage and we try to understand what you're doing around LARC, for instance, or another target in the field, can we find there all this information, or is there a kind of first request and then a process to get there?
I think there's two stages to this. Where we have generated reagents or structures or compounds or know-how that we've made publicly available, either through our website, through publications, or through other databases such as the PDB, then of course that's immediately open to everybody. And the open access part of what we do there refers to the fact that we put this out very quickly. As soon as we have it, we make it available. We don't tend to hold things back unless we have to. The other half of it is that, as Chas mentioned, we have quite a large pipeline of work that's going on at any one time that is changing on a monthly basis. We don't tend to immediately show what we're doing because it's a lot of work just to keep that up to date. Again, just reach out to us. We'll immediately tell you if we're working on it or not and who you need to talk to directly. And what I find is that we're incredibly collaborative in that context.
Yeah, I think it's really a no-brainer to get in contact and do things together. But I'm also thinking about, I don't see there are many questions, so let's just get the discussion going. So
before you go on, Bart, can I just touch on something that I think Paul said, is that when we've generated one of these target enabling packages, we actually have an external review committee that looks at all of that science and data and says, yeah, this is high quality, now you can release it to the world. And similarly, when Paul was talking about the probes, again, once we've generated that probe molecule, we again have an external peer review committee who look at it and say, yeah, this is high quality, it's potent, it's selective, it's cell active, now you can release it to the world. And I think John touched on this. What is incredibly important for us is that any tool we generate, it's well characterized, and any data we generate, it's robust and it's reproducible. I mean, you and I have chatted, I mean, in the past few years, some of our pharma colleagues have published some fairly high profile papers saying that 50–60% of academic literature, they can't reproduce it. Now, for us, that reproducibility is incredibly important, because if one of our farmers can't reproduce it, they will stop working with us tomorrow. And so we need to make sure that robustness, that reproducibility.
Yeah, I like the concepts. So how do you achieve that quality control throughout the institute or the collaboration?
I mean, Ofer, Paul, Killian, Liz, guys, John, Eleanor.
I think
why are you so good, Ofer?
Because it's even in the industry, I sometimes struggle with that type of quality, permanent quality delivery. You depend very much on your individual scientist. It's a very tough thing to have a top quality of experimentation.
Yeah, I can talk maybe from the structural biology point of view. The community of the structural biologists have instituted all kinds of quality measures. And so when you deposit a structure in the database, you are asked questions and there is a validation report that comes out. And if there are anything that's missed, and this is generated automatically, so you can't really fudge it in many ways. Of course, there are bad structures.
Biology is probably as close as you can get to exact biology, exact science in biology.
And I think I would say, maybe John would replicate that a lot of people who are working on cell biology, we have a core of people who actually worked in assays in industry and are very conscious of the standard of reproducibility that's needed in drug discovery. So this mixture of people from academia and industry really feeds up the building up of standards.
And maybe if I could comment, Bart, on the probes, I think Paul didn't touch on this. I mean, when Paul and Oleg and Killian, etc., generate one of these probe molecules, you know, they are screening it often against 600 or 700 other off targets, you know, the whole of the kinase panel, lots of our epigenetics, some GPCRs, ion channels, etc. These are extremely well characterized. The second thing is, what they've also done is they've generated a structurally similar but inactive molecule. So it's structurally similar, but it's inactive. So if you like, you've got a positive and a negative control. And then thirdly, what they've done is that sometimes they've also generated another active molecule, but it's a completely different chemotype. So now you imagine if you've got two actives from different chemotypes with inactives that are structurally similar, you can do a fairly definitive cell biology experiment and say, this protein's involved in this part.
Just if I can say a few words. We've been working with 9 pharma companies. We provided data which suit the quality of the internal database. They've been validated. We learned a lot, how to do experiments, how to set up, how to document itself. So overall, we have a very well-defined way to generate data, which will be reproduced in at least 9 pharma companies. So that's basically the training which we received in 5 years of working on chemicals.
It's a wonderful constellation you got there together. I'm just watching. If anybody wants to ask questions, please post them on the question and answer box or in the chat if your Q&A works. I really want to profit from the presence of all these people, to think together with them how we can move things forward from the DRI towards this cooperation. It would be just fantastic. It would enhance our drug discovery a lot. Do you have, I mean, how do you work together with the Drug Discovery Institute? Do you have a kind of regular meetings, or do you have a way of funding collaborations there, or do you have a kind of mutual understanding agreement? How we structure?
We'll let John and Eleanor and Paul answer that question.
Yeah, I think it's a good question. But I think for us, it started by Chas inviting us to contribute to the target prioritization meetings, which was effectively the conduit for nominations in our therapeutic area to go in. So we were able to put up our targets, and Eleanor over the years has updated that. And then we look at the where they are in the portfolio, and we obviously change our priority from time to time and encourage the SGC PIs to work on the target that's of particular interest for us. So, for example, it's no secret that we're interested in SHIP1. That was a target which OFA picked up for us. And so, you know, we're very keen on the progress that he's been making. And I think the other thing that's worked really well is together we've been able to accelerate the funding to support SGC work. So Paul's work, especially with the AMP-AD consortium, contributed towards the call being raised there by the NIH-NIA to go to AMP-AD plus. And that led then to the OpenAD call, and we all contributed into that application. And that has brought more money then for neuroscience targets into SGC Oxford. So I think that type of mechanism has been working very well.
I think you're spot on, John. You always say things so nicely. I think, Bart, I think what the SGC has done with the DDI in Oxford, we would like to do that with the DRI across the UK. So, you know, over the past 4.5 years, John and Paul and Eleanor, they've told us which targets they're really interested in. We've generated the tools, and then of course they've got the disease relevant assays, and now they're sort of taking them forward and creating packages, working with pharma, working with venture capitalists, etc. We just want to turbocharge that. You know, we built the scale and the infrastructure, and we want to take a lot more things through more quickly. Yeah. And I think we have an opportunity to do that in the UK. And I don't think that sort of opportunity or infrastructure or network or culture exists anywhere else in the world.
No, I also see an enormous opportunity here to bring two pieces of a jigsaw puzzle together. Giovanna, there was a question I've seen.
Yeah, there was a question from Yu from Imperial College. He says, often when developing compounds in a high throughput manner, we need an assay for fast readout. I was wondering about the view of activity in collaborative and novel assay development per se with the SCGC, with the Structural Genomics Consortium, perhaps as a first step to compound screening.
So who's that question? Maybe Ofer, Oleg, Killian, Paul, John, anybody?
No, I'd say from our side, we, in these target enabling packages, we typically will develop biochemical assays with purified proteins. And again, because of the experience we have with Oleg and with Paul and with the other groups, I just can give two examples of which assays that we developed went directly into high throughput screen in facilities that do that. So I think we have the experience in getting the right Z values and readouts and so on. I think the ODDI, John Davis's team, have some fantastic assay developers there, and they've moved the SHIP1 assay from a very crude initial assay to something that's suitable for screens. I would say, John, that your team, John and Elena, are really specializing in cell-based assays that involve iPSC cells or engineered cells of various sorts. In the SGC, we tend to do more generic cell assays that ask questions about target engagement. So it could be a hex cell that has some nanobread construct or some other thing that can be traced. And just to answer the simple question of what's the dose response for target engagement in cells before they go into the more expensive assays on specialized cells. But I think we've collectively have a lot of experience in doing that. And that's part of our target enabling package. It's a protocol for assay that can then be adjusted to whatever setup or throughput that people want to use.
And we've not talked about this today, Bart, but you know, I can assure you that the colleagues on the call, you know, they're producing very, very high impact papers. They're training lots of students, training lots of postdocs, attracting lots of fellowships and grant funding. So all the things that we have to do as academics, you know, these guys are doing it in spades. So I think it, you know, again, I think there's a lot of potential synergies there.
Yes. I think that's a nice note to maybe conclude our this seminar and this discussion. I thought it was very enlightening and very stimulating actually, and I appreciate a lot that you all make time to visit us basically in virtual space. I'm going to talk with Giovanna. I'm also going to take this up internally with a couple of our people walking into that direction. I'm also thinking a lot about how we can consolidate, because you alluded to that, the collaboration with the DRI, because I think the DRI, the DDI, is the natural transition between what we are doing and what you are doing. And I think it should be a natural pipeline. It doesn't have to be top down, but from bottom up, we should consolidate that and make it, as you say, UK pipeline of innovation. So thank you very much. We will take it internally for further discussion, and I'm sure this will have a follow-up in the months to come.
Thank you all very much. Bart, Giovanna, thank you for organizing it. Thank you to my colleagues for speaking, and thank you to all of you for listening.